Вот. И сегодня у нас такой традиционный формат научного клуба, то есть первое выступление у нас будет чисто научное, и, на мой взгляд, достаточно интересное. А вторая часть у нас будет такая более, так сказать, такая околонаучная, но тоже, на мой взгляд, интересная. Это будет анонс, собственно, тех лекционных и семинарских курсов, которые нам любезно предложила провести, собственно, компания Biogenic и представить новые системы микроскопии компания Andor Technologies. Вот. И сейчас я хотел передать слово Бисеру Наталье Михайловне, сотруднику кафедры зоологии беспозвоночных, которая нам представит нашего первого приглашенного лектора, профессора Свободного Берлинского университета Hans-Joachim Pflüger.
Спасибо, что все пришли и заинтересовались лекцией профессора Pflügera. Он не первый раз посещает Московский университет и не первый раз будет делать здесь лекцию. Несколько лет назад он делал лекцию о строении нервной системы анихофор у нас на кафедре. И все, кто участвовал в нейробиологической школе на Белом море в 2012 году, конечно, помнят его лекции, его занятия, поскольку он был один из организаторов и как бы один из движителей организации этой нейробиологической школы. Профессор Pflüger в настоящий момент является деканом факультета химии, фармакологии и нейробиологии Свободного Берлинского университета и является всемирно признанным нейробиологом и нейрофизиологом, работая при этом на объектах беспозвоночных животных. Он работает в основном на насекомых, это его любимый объект. И именно насекомым он посвятил большую часть своих исследований. Однако эрудиция его как ученого распространяется намного шире, чем насекомые. Он является экспертом по многим медицинским вопросам нейробиологии, также является представителем комитета по исследованию головоногих и нейробиологии в области у головоногих и председатель множества всяких комитетов международных по нейробиологии. Что для вас, для нас, для всех очень, мне кажется, привлекательно, это то, что он неоднократно участвовал в организации молодежных школ по обучению молодежи в области нейробиологии. И я думаю, познакомиться с его исследованиями нашим молодым студентам очень приятно будет. Давайте пригласим его.
Okay, thank you very much, Natalia, for your introduction. And well, this is a respectable lecture hall, and it's amazing. So, what can I say? So, I'm sorry, you have to switch to English because, unfortunately, I'm not able to speak Russian. So, I will talk a little bit about biogenic amines in insects, and I mean, what are you doing today? You go to Wikipedia, go to the internet, and you point in biogenic amines, and then you find a list of indeed biogenic amines, which are relevant for humans, as relevant for mammals, as relevant for invertebrate animals, in fact. And if you would read all of those up, please don't, then you would see that there are lots of words that are common to behavior, common kind of expression, reward, addiction, behavioral reinforcement, and so on. I am studying this one, tyramine and octopamine. And the name says already where this amine has been discovered. It was in the octopus, in the salivary gland of octopus, and it turned out it is an amine that is widespread in the animal kingdom. So, what are we talking about? We talk about the original substance is tyrosine, and then from tyrosine you synthesize either the catechol amines, which are present in us, for example, or you synthesize the phenol amines that are present in the invertebrate animals. Of course, there are amines that are also present in the invertebrate animals, but as far as one knows, I mean, there is really not really good evidence that there are noradrenaline or norepinephrine in invertebrate animals. But if you look at these two, for example, tyramine and dopamine, you will see, I mean, they are structurally very, very close. The only thing is that actually dopamine has a hydroxyl group more than tyramine. And the same would be if you would compare dopamine to noradrenaline. So these are structurally very similar molecules, and I'm going to talk about these two. So, one of them, well, you all know about plagiarism. You shouldn't do that, but certainly one of the most copied sentences from, I think, an original review by Peter Evans in 1985 was, dopamine is released as a neurohormone, a neuromodulator, and a neurotransmitter. And I did use this phrase as well. So, what does it mean? First of all, it's clear what a neurotransmitter is. So a neurotransmitter is a point-to-point connection. It's a clear, classical synapse with pre- and postsynaptic specialization and with ionotropic receptors to allow a fast action. But yet one of the things is already that you may have metabotropic receptors for the same transmitter that may act on this in a different way. So either it may change the action of the classical neurotransmitter, or actually it may in any way change the response to the neurotransmitter if there are metabotropic receptors. So you see there's already maybe something which you associated usually with neuromodulation. So neuromodulation, of course, is a particular neuron. This would be neuromodulation if there are metabotropic receptors that would not allow an independent modulation. Because there's only one neuron, and if you have a metabotropic receptor, depending perhaps on the concentration of the neurotransmitter released, you would have automatic modulation. You could not control this from a third or outside neuron. But with neuromodulatory neurons, of course, you can control this. And in general, you have a different type of synapses. You have what is called en passant synapses, and they release their, and you do not have, as far as one knows, postsynaptic specializations. So they release their transmitter into what actually was put forward by a Russian neurobiologist a long time ago, into a volume space. So you have a certain volume space that may be depicted by glial borders, that may be depicted by tracheal borders, that may be depicted by up to now unknown borders, but nevertheless, everywhere where you have receptors for your neuromodulator, you will have action. But still, it is a point-to-point connection, because the neuron clearly has an axon and innervates all these different target organs. And these target organs could be muscles, could be glands, could be sense organs, and so on. So, then, of course, you see, these neurons could also have, on their way to their outside, to their peripheral targets, some release sites for the neuromodulator, and then this neuromodulator would automatically be a neurohormone, because it is released into the big hemolymph space of an invertebrate animal, particularly of an insect. And this is, of course, typical for a neurohormone, because you have paracrine release, and that will then, wherever you have a target cell with a receptor for this neurohormone, you will change this particular cell. Okay, so what are my aims? It's actually to describe the behavioral action of biogenic amines, such as tyramine and dopamine, at a behavioral and cellular level. So, in particular, identify the neurons, so we use immunocytochemistry, characterize their patterns of activity, when are they active, under which conditions, then characterize wherever there is a network, and then make comparisons by studying different, for example, closely related or more distant animal species in order to find out whether there are some similarities, and then define possible evolutionary building blocks of neural networks and behavior. So that's our aim. So these are the animals we use. So we use, at one time, of course, large and big animals, the locust as a hemimetabolous insect, then the tobacco hawk moth as a holometabolous insect, and of course this is also, of course, a hemimetabolous insect. It's a stick insect. From time to time I go back to this animal because this is the one with which I did my PhD thesis. So I have some kind of hidden liking of this otherwise pretty boring animal. And is the fruit fly. So this we use, of course, as a genetic model. And we should not forget, some people may forget it, but fruit flies are insects. Okay, they are not somehow vertebrates, but they are insects. Okay, so the neurons which release tyramine and dopamine fall into, don't read all these numbers, fall into principally two categories. And these are the ones in the CNS, which means the brain and the ventral cord. And these are actually in the brain. So you have a number of dopaminergic and tyraminergic cells, and they are all paired. So they are all symmetrically paired, as you would expect from a bilaterally symmetrical animal. And then in the ventral cord, there are a few of those, but not many. In the ventral cord, however, you have lots of them of those. So they are all midline neurons, and they all have bilaterally symmetrical axons to innervate, for example, if they run to the periphery, the same targets in the periphery, or if they do not run to the periphery, at least they either go up to the brain or they descend the whole ventral cord. And I will show you some examples, which are, of course, not from my own work. This is my colleague Peter Bräunig in Aachen. And these are neurons in the subesophageal ganglion that innervate basically all principal neuropilots of an insect brain. And there are quite a number of those. And then you have a population of ones that descend in order to supply all the major neuropilots of the thoracic and abdominal ganglia. Then you have some in the brain that form enormous innervations of the optical ganglia. And as it is known now, this octopamine helps to actually modify the responses of interneurons in the visual ganglia that completely differently shape the characteristics of a fly when it really actually flies. So you need octopamine for the proper activation of these neurons. And these are actually neurons that we found. So it's a population of paired cells in the brain. And these ones are, this in green is the same cell cluster with a tyramine antibody, and the one in magenta, and this shows you better here, are neurons that you can fill by doing what we call a backfill. So you cut a connective, you put in the dye, and you let the dye diffuse to the brain, and then you will label all the neurons that have an axon that you just cut. And so these cells in magenta all have axons in that connective. And you see, as we use also the tyramine antibody, where the two colors are in the same cell, these cells are white. And clearly, so we have a population of neurons that descends to the actually ventral cord. And one of my PhD students, Sergei Hartfield, he is in his PhD thesis looking at these cells, and this is one fill of such a neuron. And you see they descend the cord. This is the subesophageal ganglia, the prothoracic ganglia, and it turns out these cells collect mechanoresensory information from the whole body. So if you touch the whole body with a brush, these cells will fire. And the pattern of kind of firing is different in these maybe different cells. So that's not really clear yet. I mean, he hasn't finished his PhD yet. But anyway, so these cells, which I was talking about, are collecting mechanosensory information. So you could say, well, if an animal is aroused by touches or by sensory stimulation, then these cells definitely will fire. Now, why is this interesting? It is interesting because these cells were found to produce octopamine only after stimuli that we would call a stressing stimulus. So we could only identify them as having octopamine or synthesizing octopamine after we stress the animal. If you don't stress the animal, you will not see octopamine in these neurons, only after stress the animals. And a recent paper was very in favor of this, of a French group, because they showed that in the cockroach, it's the enzyme that synthesizes octopamine from tyramine. That's tyramine beta hydroxylase, TBH. That is the crucial enzyme, and that is actually regulated by stressing stimuli. So you have an enzyme that you only activate when the animal experiences stress, and then these neurons make octopamine, and then they are active. Probably because stress involves some kind of arousal mechanism, and that way they will discharge octopamine in the ganglia. So that's an interesting kind of group of cells to work with. Then there are these descending neurons. This is one of them, and you see it's this population here in the subesophageal ganglia. So basically, in the ventral cord, we have a few of those paired cells, but basically all the other cells are unpaired median cells, as they are called. And when their cell bodies are dorsal, they are called dorsal unpaired median cells. When their cell bodies are ventral, they are called ventral unpaired median cells, and that's why they're called dumb and lump cells. And tyramine in the ventral cord, well, a few, but very little. The main source of tyramine is actually the brain. So, now you see these cells. So this is an antibody against octopamine, and you see a whole mound of a meso and a metathoracic ganglion, and you see the whole cluster of cells that are octopaminergic. And this is in the subesophageal ganglion, and when you make a really sagittal sections, you nicely see this is just a one layer of cells, and so they have all this nice path, so it's all defined, of course. And here you see some of on the ventral surface. But if they are on the ventral surface, I mean, their neurite, as it is called, strictly goes to this thing, and then they do exactly the same thing as the dorsal ones. So the idea is why they are ventral cells, at least in the locus, is that in development, this is the metathoracic, this is the first abdominal neuromere, the second abdominal neuromere, and the third abdominal neuromere. So the ganglia for the first three abdominal segments fuse. And if in this fusion process, which is of course a dynamic process, for some reason or the other, a cell body gets misplaced by the growing neuropile to the ventral surface, it will just stay there. No chance to go back to the dorsal surface. So instead it grows a very long neurite until it reaches the point where it should bifurcate to make these two axons. And I mean, to prove this, we have actually metathoracic ganglia where you have only one and two dorsal cells, or you have three dorsal cells. We never found three ventral cells, so you have different complements. But that happens only in these fused kind of ganglia. So, unfortunately, these films don't work as I actually wanted them to work. So, this is one of these cells which we injected, and then you just see how nicely, so cell body, neurite, and then there are these secondary neurites, and this is just not working really properly, but it should go in the fourth circle. But you see, if you do these kind of new, nice 3D reconstructions of neuropiles and ganglia, you can address the neurons into such a, it's not really a standard one here, because it's only 4 ganglia fused in order to give this kind of standard, or an average. But you see how nicely these branches fit into these patterns, the architecture of such a ganglia. I mean, this is what nowadays is kind of a standard in neuroanatomy. So this shows you the different types that we can identify. So these are all, it's this you saw in the immunochemistry part, you saw these nice clusters, and these clusters are kind of, so each of these ganglia has this cluster, but here are the different types just indicated by colors. So you see some of these cells, the axon actually trifurcates, and you can imagine that there are lots of target organs of these ones, you know? Whereas some others, like here, they only have an axon through one of these nerves, and I mean, they innervate, of course, much less targets less target tissues in the periphery. So these are just the different types that we can find. And you can, of course, do also very elaborate reconstructions. These are then automatic reconstructions. Now, then you have digitized all this, and you can then do all statistics that you want. How many branches, what order of branches, and how are the differences between these cell types. And so there are clear differences between these cell types. For example, if you take these ones, they are missing all these parts of branches. You see, so they do something particular. And actually, these is the population of cells where we don't almost nothing, because they are very difficult to get, and they are very, very silent if you work in such an animal that, of course, in order to record from these cells, you have to open, you have to have it stationary, and the animal has to cooperate and still do something, because it has to be alive. So these cells seem to do, under these conditions, very little. So these are then these different types, and I have summarized for you what these different types do. They fall into more or less three categories. There is one group of these cells that innervates wing muscles that are used for moving the wing up and down. So these are called power muscles for the wing beat. And then there is a group, they innervate all leg muscles, they innervate lots of the thoracic muscles that are not wing muscles, so they're not associated with the wings. And then you have a group that innervate the visceral organs, like the oviduct, the spermatheca, the insect heart, the gut, and so on. So that they obviously call them the visceral group. And how are they characterized? So the interesting thing is that you can find out which cells are talking together. So when you record from pairs of these cells, you soon find that they either are paired, so they receive common inputs like here. So these are two completely different cells, because in order to be sure, you have to fill the cells and stay in the cell, and then you of course see the two kind of cells that belong to this type. You see, this is a cell in the mesothoracic ganglion, this is a cell in the metathoracic ganglion, but you could see them as almost one cell, except the one is spiking here. So one spikes here, the other one doesn't, and then this spikes and the other one doesn't. This spikes and the other one doesn't. But still, they receive really a lot of common inputs, where these two cells, they do not receive a common inputs. So by making lots and lots of these recordings, you really can find out how the network is wired. And we do know that actually the source of these common inputs is the subesophageal ganglion. You can cut away the brain, these will still survive. So the really motor and drive for these cells must reside in the subesophageal ganglion. And then, of course, you can define stimuli that will drive these cells. And there's an interesting thing which we would never have thought. So this is the network. And I only would show you what is in red is neuromodulatory, what is in black are motor neurons and sense organs. And so you have in the subesophageal ganglion, of course, this is the motor command center. So if you have an animal where the connection between the ventral cord ganglia and the subesophageal ganglion is cut, this animal would never ever walk. It will be able to make one step if you stimulate the leg, but that's all. However, if you cut the connection between the subesophageal ganglion and the brain, which would be the supra-esophageal ganglion, then the animal walks uninhibitedly. You cannot stop this animal. It will just walk. So that shows you that for maintenance and for keeping this kind of coordination, you need the subesophageal ganglion. And so that's an important motor command center. And so if you look at this, so you have local neurons, that has been worked out beautifully by groups in England, by Cambridge, by Marco Burrows and many others. And so you see there's these pools of neuromodulatory cells, they all innervate the same muscles. But now if you go to feedback, you see sense organs monitor, of course, the action of the muscles. And if they go back to the motor neurons, we have monosynaptic connections. There are, of course, quite a few monosynaptic connections, but the majority of connections goes via the interneural network. So they are polysynaptic. And we don't really know about these connections. Now, if you go to the feedback to the neuromodulator neurons, you see there is absolutely no. So this is a big myth still. It's a black hole for us. We don't have any direct sensory input to these neuromodulatory cells. We have inputs polysynaptic. That we do have, but never we could not identify any direct input. So the idea is that actually maybe the only inputs come from interneurons, or then you see the major surprise is it goes all via the subesophageal ganglion. So if you have an abdominal sense organs and you tickle the hairs there, of course you will get a response in one of these neuromodulatory cells. Then when you cut off the subesophageal ganglion, this response is gone. It's no longer there. So that means all sensory information somehow is collected by neurons in the subesophageal ganglion, processed, and then is used to actually control these neuromodulator neurons. So that would be one of the predictions which we made. Okay, so let's move to the peripheral aspects of these neurons. So this shows you how an insect muscle is actually innervated. So in green, we just made a stain of the motor nerve that innervates in this way. It's a muscle which controls the twist angle of the wing, either whether it's pronated or supinated. And this shows you the innervation. And by using an antibody against a synaptic protein, synapsin, everywhere where it's white, you have a synaptic site. You have a neuromuscular synapse. And then, of course, you could use antibodies against tyramine or dopamine. They are a bit tricky to work with, but in the end, it works. So here you have anti-tyramine and you have anti-synapsin. So it's all double labeled as well with synapsin, but very little. But these are very nice. And these are the neuromuscular synapses, and these are these en passant synapses by the neuromodulatory fiber. And if you do a sketch, it looks like this is the motor fiber in green, and what is in magenta winded around this. This is the neuromodulatory fiber. And for example, here you have the neuromodulatory fiber in magenta, and how it actually is related to the normal neuromuscular synapses in green. So you get an idea how this looks in the periphery. And this actually shows you the octopaminergic terminals, all these varicosities, these sort of type, as in Drosophila terminations called type 2 terminals, whereas usual neuromuscular synapse is called a type 1 terminal. So if you then take the antibody, you see this is anti-tyramine and this is anti-octopamine. So you see, indeed, in these terminals, you synthesize also octopamine from tyramine, so it's present, and the enzyme, of course, has also to be present, otherwise you could not do this. And so with Natalia, we even did the immunogold methods, and maybe you can see it's also pretty tricky, but you can see some gold, we have a few others where then you have the typical for amines dense core vesicle with immunogold precipitation of anti-octopamine in these immunogold, in these dense core vesicles. Now, if you, then of course, as we want to use Drosophila as a genetic model, we switch to Drosophila, and in fact, so we make a transgenic animal where all tyramine and octopaminergic neurons are labeled by GFP, by green fluorescent protein. And this shows you the larvae, and this shows you the adult. And in addition, we have a marker for another synaptic protein. So whenever this is Bruchpilot or NC82. So wherever you have the synaptic sites, you will have a label. So it nicely labels the neuropile, the outline of the neuropile. And then you also have some marker in blue, which gives you, in general, a marker for neuronal tissue, because it's an antibody against horseradish peroxidase, which is known to label epitopes in the cell membrane that only neurons have. And these are, of course, the cells. You see already, again, the bilateral symmetrical axons, so that is as predicted as you see in larvae. It's exactly the same kind of thing. But now you have transgenic label, and you see all muscles in that Drosophila larvae receive this wonderful kind of innervation. Here you see particular one, and these are these beaded fibers. So these would be these type 2 terminals. And of course we can go further. We can also use antibodies, and you see all these beaded terminals are full of tyramine and octopamine. So this is exactly the same thing in Drosophila. So I told you it's an insect. And here I think this is a Drosophila muscle. Here you see the muscle fibers, and you see one of these varicosities with the dense core vesicle, and definitely you see some immunogold staining in these fibers. And then you can take different combinations. So this is our transgenically labeled green fluorescent tyraminergic octopaminergic fiber, and this is the antibody against Bruchpilot. So everywhere in a synapse, in a neuromuscular synapse, these dots would label the active zones where vesicles can dock to the membrane and release the normal classical neurotransmitter. And if you see now, these varicosities have only one dot. They only have one active zones, and actually in many of the previous, it was very difficult to see. So many people have thought, well, they don't have this active zone, but I think they really do have these active zones, and this becomes really clear when you use other labels. For example, here we use NC82 and HRP, and you see nicely, this is the transgenic, the octopaminergic fiber. You see these wonderful varicosities, and you see here dots in every of these fibers. And this is the motor fiber, and you see also the synapsin fibers of the release sites. So that means that these type 2 terminals use the same synaptic or very similar synaptic machinery to release the vesicles, but just very, very little active zones in such a varicosity. Okay. So, let's just look at neurohormone versus neuromodulator. So it has been shown a long time ago that if you have an animal that actually flies, or before it flies, and when it flies, you can measure the hemolymph content of octopamine, and it turns out octopamine is very high in the beginning and then reaches a steady level before hormones kick in, and these are the adipokinetic hormones, AKH. There are several ones of them, AKH1 and AKH2. So octopamine is kind of the first source of providing the animal with energy that it definitely needs for takeoff, because takeoff is like in any airplane, takeoff is an energy demanding behavior. So, of course, then it has been shown that octopamine has hyperglycemic action. So if you inject octopamine in the hemolymph, suddenly the amount of trehalose, which is the insect glucose, so to speak, goes up, and also the lipids from the fat body are broken down, and you have fatty acids in the hemolymph. So octopamine acts on the cells of the fat body, that has also been shown before. And the interesting thing is, a recent paper, it acts also on the tracheal cells in order to somehow make gas exchange more efficient. And this is exactly what you require when you need energy and breakdown of lipids. You need a lot of oxygen, and so you have to make also oxygen exchange, gas exchange more efficient. And so these things in the hemolymph nicely go along with the action you would actually follow. Now, where is this octopamine coming from? And if we also look back into a very old paper from Peter Breuning, and that's actually also what we have seen, some of these bilaterally symmetrical axons, which you see, they suddenly have axon collaterals, that very tiny ones, that run to the sheath, to the surrounding glial kind of barrier, which surrounds every nervous system, even in insects, and form these, what we would call a neurohemal release site. So they have lots and lots of varicosities, and they dump octopamine into the hemolymph. And it turns out these are the neurons that are the most sensitive to any sensory stimulation. So they would be in a perfect position to actually increase the octopamine content in the hemolymph. And then there are other organs, and that's a master thesis in my lab at the moment going on. And she managed actually to show the release sites of what is called the antennal heart. And the antennal heart is a big muscle, well, it's a tiny muscle, but with big innervation from octopaminergic fibers. Whether it's octopamine is not clear yet, but definitely it's tyramine. This is shown tyramine. And this is the release sites of this just backfill from this neuron that has a cell body in the subesophageal ganglion. So this could also be a very rich source of octopamine in the hemolymph. So that's neurons, definitely neurons that could fulfill the hormone role of octopamine. So first summary. So peripheral automatory functions, these neurons, and that's now I think a very interesting connection to the metabolism, because they exert a neuronal control over the energy metabolism, which is called catabolism. So they may even be in a position, that we don't know, speculation, to switch from a carbohydrate metabolism to a lipid metabolism, because this is exactly what happens in these large and big insects. And so octopamine would increase carbohydrate metabolism because it boosts up glycolysis. And in accordance with this, there is one population of dumb neurons, of octopaminergic neurons, that actually, to our big surprise, is switched off when the animal flies. And it is exactly the neurons that innervate wing muscles. So, and these dumb neurons, to our, again, surprise, are active when the animal is resting rather than flying. So what happens is that these animals produce a metabolic signaling molecule, which is called fructose 2,6-bisphosphate. Everyone who has to deal with glycolysis and the tricarbon cycle knows this substance. So in any muscle, rat muscle, or here locust muscle, octopamine would boost up glycolysis. And that's, so insects make no exception to this. And so what these neurons do, they produce a lot of this signaling molecule, which is not used in a resting muscle. But as soon as the animal flies, and this muscle needs ATP, then glycolysis, as a first start energy supplier, uses this method to supply the muscles with the take-off energy, and then later this is all done by the adipogenic hormones for these long periods of flight. So we would speculate that this is only to regulate the take-off energy that you need for these heavy insects. And if you now look to Drosophila, these we never found any population that is inhibited by flight there, because Drosophila never use lipids for flight. They only use sugars for flight. So they need sugars all the time. And indeed, these cells do actually, they are active while the Drosophila are flying. So I think the dumb neurons and dumb neurons are powerful regulators, controllers of metabolism. But in addition, they have, of course, all effects that help in this. So they also make a neuromuscular transmission more effective. So they change the efficacy of the normal, classical neuromuscular transmitter. Okay? And also, they have this neurohormone action in the hemolymph. So they also use this kind of energy metabolism. And then what happens with the visceral muscle? Indeed, like in our vegetative or autonomous nervous system, all visceral activity is switched off. So if you have these neurons that innervate the visceral organs, they inhibit myogenic, for example, rhythms, except one visceral organ, and this is the heart. The heart is accelerated. But oviduct and so is all shut off, gut is shut off. So to speak, this is the autonomous nervous system of insects. Because even, makes sense to do this, because even an insect, when it flies, doesn't like to lay eggs. So it shouldn't do this, okay? So this is exactly what these neurons then achieve. Okay, so in short words, repair skeletal and visceral muscles and other target organs for soon to come dynamic action such as locomotion or flight. Okay, then the remainder of this, I would just point out to something which has just been appeared or recently appeared, and this is that these people have looked at these axonal terminals of octopaminergic fibers, and they have found a remarkable plasticity. So if you have a larvae of a Drosophila, you let it hungry, so you don't feed it, for 3 hours. These axonal terminals will sprout, they will develop what they call synaptopods, they will form more axonal release sites, and they will increase the size of the neuromuscular synapse. So to speak, to get the final thing out of a muscle when the animal is hungry, is starved, and has to actually then fulfill a new task. And what is the new task if you're hungry? Yes, you have to locomote. So all these starved animals have an enormous drive for locomotion. So they locomote much more than a satiated animal. And it turns out that there are octopamine receptors involved, and the other receptor, which is then involved in stopping too much growth, sprouting of the axonal fibers, is an octopamine 1, a B1 receptor, and this is a B2 receptor. And so the balance between these two octopamine receptors will decide how much new synapses you will actually make in the system. So these are results from nature. And here you see some of these synaptopods, and well, this is just from their paper. So let's go then to Drosophila. So I don't know whether this works, probably not, because then this animal should fly. It doesn't, but I mean, so here you see the hook, where this is hooked up and it stationary flies. And then you can measure wing beat frequency with a high-speed camera. You can measure how long it will fly, how many times you have to blow on it before it starts to fly again, when it stops, and so on and so on. You can all measure these. So we do a very simple behavioral task. So how does a mutant fruit fly, Drosophila melanogaster, that cannot produce octopamine? So you can make a mutant that cannot produce octopamine. How does it fly? Can it fly? Or can it not fly? Or what happens? So I don't want to explain everything to you, but you see these yellow bars. These are the normal. So that's the normal variability of not really selected fruit flies, just taken out from the stock, and they are, of course, they feed as they like, and these ones are the mutants. So I mean, you see immediately that the mutant can fly. So these animals can really fly, in contrary to what we expected, but they have no stamina to fly. They just can't fly long enough. So that's the result. They can fly, but they do not have the energy or the stamina to fly for a long time, and that has now been shown for many other locomotory behaviors. For example, the jumping. They cannot just jump as high or jump as frequent, the mutants, as a normal wild-type animal. So now we can, of course, say, and these are very recent results, we can say, well, we know now that these axon terminals of octopamine in the periphery, they also have this Bruchpilot, this active zone proteins. So what happens if we make an animal where this active Bruchpilot protein is not functioning? So we can interfere with this system, and if we do this, indeed we can use RNAi so that this Bruchpilot is not formed in these animals, so that they cannot release if this is really crucial for flight. And you see there is a big difference between this is the kind of animal that has no release, and these are the normal ones, the wild type, and this is the wild type in this background of this, where this actually animal was generated. So, expression of Bruchpilot RNA in optogenetic and pathogenic cells led to a decreased flight duration. So exactly this is what we have shown in the behavioral experiment. So indeed, for doing this, apparently you need this octopaminergic control for energy metabolism in these insects. So let's see what actually you now can do in a behavior in pharmacology. So what we take these animals, and now we want to see, well, pilocarpine is a muscarinic acetylcholine receptor agonist, and is widely used in insects for releasing motor patterns. And so we use this as well because it works absolutely perfect. So if you have a completely isolated nervous system, ventral cord of an insect, the insect as a body has long rod, it's a big out nervous system in a petri dish, and you put on pilocarpine, this animal will, what we would call, fictively walk. So it will produce a walking pattern that is similar to what you would observe in a normal walking insect. So this is why it's called fictive walking. And fictive locomotion is a big thing in neurobiology because it can produce by fictive flight, fictive swimming. I mean, even a fish, a vertebrate, produces an isolated spinal cord, produces a perfect swimming pattern, and it's called fictive swimming. So you don't. So vertebrates are no exception to this kind of approach. So let's see what happens. So this is what we have. We have this time is the tobacco hawk moth. So we have an isolated meso and metathoracic ganglia. We have only these two ganglia, and then we put on pilocarpine. And then we record from these muscles. So these are wing muscles, so they would indicate flight. That would be an indicator of flight. And we record from these muscles, so the stump of these, no, not muscles, but the stump of these nerves. We suck it in. in an extracellular suction electrode, and then you can nicely extracellular record the pattern. Of course, I mean, you have to interpret these patterns, and you can only interpret these patterns when you have carefully studied a normal intact animal. So only by doing this long and many times before we can interpret these patterns. But all these spikes, you see, these are single motor neurons that discharge and that are identified. Now, the interesting thing is that under certain conditions, pilocarpine produces something which is never ever seen under natural conditions. And that means this animal flies and walks at the same time. I should say fictively flies and fictively walks at the same time. Because usually if an animal has tarsal contact, flight will stop. And at the same time, if you lift an animal and has no more tarsal contact, it will start to fly. So these are two exclusive behaviors. But under these artificial conditions, you can release both pattern generators, both central pattern generators, and of course now you can do all kinds of things. Well, do they influence each other and so on? Actually, I don't want to go into detail, they don't influence each other too much. Very little overlap between the pattern generator of walking and flight, and this is why you can get, here you have a flight pattern, and here you have a typical walking pattern. Now, if we, however, then use tyramine, which is the precursor of octopamine, well, it exactly does the same. Right, the flight looks a little bit different, but still, this is flight according to all the criteria. So, tyramine also evokes flight and walking, whereas octopamine, we never, well, maybe 5% of the preparations, 5 from 100, would also show walking related to octopamine. But octopamine is really the stuff that makes the animals fly. It's not the transmitter of the neurons that form the network, it is only a modulator, because transmission between the neurons is most likely cholinergic. And there are lots of pharmacological reasons to believe this. So tyramine and octopamine are modulators of this cholinergic network. And so this gives you just so here are the incidences of flight, the concentrations, the incidences of flight, of walking, and here of flight. These may make you suspicious, but I can assure you, insects have a wonderful blood-brain barrier, and it's very efficient. And you, in order when you have such an insect nervous system that you have not sheathed, that still has the glial barrier intact, you need high concentrations of these drugs. But you see, tyramine in low concentration and in high concentration releases walking. So these are the black bars. And in a higher concentration also flight. Octopamine, this was pilocarpine. So this is pilocarpine. Tyramine does exactly the same thing. So low and high concentration, this is walking, incidences of walking, and this is incidences of flight. So in higher concentration, it evokes walking and flight. But octopamine were a little bit, but this is just flying, and this is just certainly not walking. So that's really what we would then say. So there are concentration-dependent effects, and so we would say that probably it's always the balance between these different substances that produces then the behavior in question. So the last point, what happens if we now use tyramine and octopamine on locus flight? So that's what we do. We release flight by not octopamine itself, but by chlordiniform, which is an octopamine receptor agonist, and works much more potent than octopamine itself. It just binds so nicely to these receptors, and you need very low concentrations and actually to release light. So wherever you use chlordiniform, the animal will fly. And then we use tyramine in addition. So we just put in more tyramine, And now to our surprise, see what happens. So here you see the elevators and here you see depressors. So here we have a nicely flight pattern, elevation, depression. And see what happens if we increase the amount of time. The depressors kick in much more than before. The overall rhythm is not changed. It's absolutely kept. But there are many more neurons now for the tyramine sort of motor, the depressor motor neurons are more active. The elevators, just like here. And now if we use a blocker of tyramine receptors, yohimbine, then we take out the depressors. Whereas the elevators just fly like nothing. So that would lead us to the conclusion that the receptors for tyramine or dopamine are differently distributed in such a central network. And for example, the depressor neurons, they should have tyramine receptors, whereas all these others here have, of course, dopamine receptors because it can also block by the respective drugs that go for dopamine receptors. So this is, I think, one of the first kind of examples where you have a differential effect between tyramine and dopamine. And as we do know that there are tyraminergic neurons per se, that up to now we never found any behavior that would make you switch them to dopamine. We think that tyramine, of course, is also a transmitter that is used as an independent transmitter. Okay, so the summary, this is the central modulatory function. So I think these central pattern generators function with cholinergic mechanism as neurotransmitters. But then if you use modulators in a low concentration, you always really walking. In high concentration, they go up then flight, and dopamine always makes flight. So this, if the animal dumps dopamine, then it will actually fly. So the overall resume, and then so you have biogenic amines such as dopamine orchestrate behavior. Now, if you just remember all what I've said so far, they coordinate, and that is the interesting nice thing, all physiological and all neuronal processes. So they're just like an autonomous nervous system of an insect. So they react to particular behavioral conditions like hunger or stress. They coordinate the peripheral system with respect to energy demands. They act on sense organs and make sense organs much more sensitive to sensory stimuli, something which you need for arousal. Then they coordinate the central networks. So they make the release, for example, of a locomotory network much more easier. So this behavior is actually generated. And they organize what we would call meaningful behavior. So if an animal is hungry, it has to locomote, it has to forage. Indeed, in foraging bees, in foraging other ants, dopamine concentration in the hemolymph is high. And they also should be more aggressive. Indeed, aggression in crickets, in flies, and other animals where this has been measured goes enormously up in an aggressive animal. And then, interestingly, and this is another similarity to the vertebrate, even the human mammalian system, the same amines are also used as modulators involved in learning, in conditioning. So, dopamine and dopamine in associative Pavlovian conditioning, and dopamine also in aversive conditioning. And so, perhaps this points out that if you are hungry, then you find food. This is a kind of reward, and then you use substances that actually make you go. You also use as the reward transmitter or the reward modulator in learning paradigms. And if you look at this is probably a similar sequence of events, I think, in most invertebrate animals. And if you replace dopamine with another amine of the biogenic amine family, then you could also solve in the vertebrate and mammalian field. So, I have to acknowledge a lot of people, and I don't read all of those. I would actually point out these are all the funding agencies that gave money for my work and for my many co-workers. And so I'm especially thankful for the German Research Foundation that supported my research ever since I was a young postdoc. And so that's a long time ago. So, so since then I enjoyed the benefit of funding for my research. So thank you very much.
Questions?
Some more details about these neurobots that can affect more muscle contraction. You said neurons make more synapses in size.
Yeah.
After it starts to do it, does it become smaller again?
Okay, well, I mean, they just showed, so they didn't do that kind of experiment, but what they showed, if they would make a mutation where these octopamine 1B receptors are not there, so you can also have a mutant animal that lacks the octopamine 1B receptor. What happens? The axon terminals of these octopaminergic fibers grow everywhere. They completely overgrow the muscle, as if they cannot stop. So that's the conclusion. If you do this, then so you require a balance between these octopamine 2B receptors. Now, how octopamine 1B receptors are stimulated by if the animal is satiated, because then this should happen. I don't know. So I'm sure there is one pathway that will exactly do this. So when you lack the amount of octopamine, then they will never be activated, and then probably in the cell you activate the signaling chain that will exactly stop growing. I don't know the events, but that would be the speculation.
Thank you. More questions?
Did you see any synapses in the brain also, or only in the neurons?
Oh yes, they are also in the brain. So as octopamine and dopamine is involved in modulating the mushroom bodies during learning, these endings of the octopaminergic, you saw one of the pictures, they are like so. But nobody knows about this volume space in the brain, because the neuropils are normally densely packed there, and nobody knows how many kind of, for example, canyon cells in the mushroom body are surrounded by such a barrier, or whether, because they are so densely packed. So that has all not been worked out yet. But definitely there are other substances.
And how to solve about that? Then the insect is walking, and then insect is flying, so neurons work differently. So I can understand how you managed to fixate the insect is walking, but how did you manage to fixate the insect that is flying?
You mean, well, I mean, an animal would easily fly, you just need a forceps. If you get hold of a locust, you take a forceps and you clip the pronotum, and then you hold it in the air and you blow on it and it will fly. So you just have a stationary animal. You just glue something to its pronotum like a harness, and you keep it stationary, and then you need a hairdryer. Okay, you switch it on, and as long as the hairdryer is blowing, this is your poor man's wind tunnel. Okay, it will fly. You don't. I mean, of course, people have made enormous wind tunnels and have measured insect flight, and you can even have the wind tunnels where the animal is on a kind of like on a leash, but it can maneuver quite freely before the wind tunnel, But you don't have to do these elaborate things in order to make the animal fly. You just glue something to the pronotum, you keep it up and switch on your hairdryer, and the animal will fly even for an hour or so.
I have more technical question. What kind of methods for imaging did you use in your experiments? I've seen the imaging, I think, confocal electron microscopy, wide field microscopy. Laser scanning? Or another techniques?
No, this was just a photo scanning microscope, like an old Leica, pretty old. Pretty old, yes. I mean, you just have different fluorescent dyes, and you have your different methods.
Okay, so I want to say thank you more for the brilliant talk and for this. As for me, I'm a molecular biologist. It was a really surprising experience about finding the latest spinal cord and walking and flying. It's pretty cool. And so,
So, first of all, I think if you want to become a scientist, you should be very curious, and you should always follow your own ideas and do things what you like. But I will also tell you that even with all the things that you like, there are aspects that you hate. So this is normal. Okay, the second thing is that I think you should you should be critical, especially if you have to rely on knowledge from textbooks and the internet. So you have to be critical about the sources. So always make sure whether you can trust the source or not. And that is very important because you have to, I think you have to use the knowledge of other people, and you have to be sure that you can trust this knowledge. And I think if you want to ask me why I did neurobiology, I would say I find neurobiology absolutely fascinating because it is studying the nervous system. And the nervous system, I think, maybe has been invented in evolution only once. And so looking at the nervous system of animals and about their circuits, about their networks, will, I'm sure, tell us something about why our networks are as they are. Because they didn't appear from new, they were shaped by evolution. And this is why I like to study the nervous systems of animals. So, okay, Dr. Pflüger for Scientific Club.